Good afternoon. It's late in the day, so I'm going to start with a bit of a spoiler. So in my view, the most interesting thing about my talk is on the first slide, and that's actually that there's a physical location, and that we're all here, and I'm here, and it's extremely exciting. And on top of that, what a location. I love this building, and after 2 years in beautiful Europe, it is so nice to be back in London and actually really feel at home. So thank you for the kind invitation. So we work on various layers, omics layers, as you call them, so ranging from the genome to kind of what we refer to as the phenome, so the largest collection of different diseases we could get, ideally from electronic health records, from medical practice. But today is a kind of proteocentric talk, so I'm going to mainly focus on the proteome. And what I try to do in the time that I have to talk about proteogenomics specifically, so here I'm not a clinical pharmacologist, but actually many of you may have an interest in specific genes, specific proteins as drug targets, and so the idea of proteogenomics is to give you some idea about the causal link between genes, proteins, and diseases, as well as the specificity of those associations, which is very important to know and understand if you're considering interfering on that. So as I said, the possibilities for proteogenomics to identify causal genes and mechanisms, to identify shared disease causes, and I'm not going to talk much about platform synergy, just point you to some of the work we have previously done. So why the plasma proteome? So as you all know, it has the proteins have key roles in diverse biological processes. They're dysregulated in disease and the most important class of drug targets. Because of the huge dynamic range here in plasma that really ranges here from albumin and functional plasma proteins down to signaling proteins, interleukins here, no single technology can capture this huge dynamic range at the moment. And then one consideration, if we take the plasma proteome and measure it, obviously the plasma connects all different kind of systems in the body. What do we actually measure if we take this snapshot? And now with broad capture proteomic technologies, we're able to do this. And what is it that we find? So the vast minority of the proteins that we detect are actually actively secreted and supposed to be there. So coagulation factors, cytokines. Most of them that we see are products of cell leakage and turnover, obviously, so they come from different sites in the body, and we don't necessarily immediately know where they come from. And also soluble fragments of membrane proteins that can be detected. So it's important just to consider what we actually find when we take a sample. And I should say, so I'm a genetic epidemiologist, and as you heard earlier in the nice talks, the effect sizes are small, and we generally need very large populations. So as an epidemiologist, we deal with those, and hence that really affects our choice of matrix. So the fact that plasma is relatively easily obtainable in large numbers is convenient, and that's why we, in most of the studies that I have experience with, that's what we've done. It also affects the choice of technology, because the measurements have to be relatively fast and scalable, and that affects in turn the content of what we get and how specific this is. So in other words, when we start out, our approaches are fairly broad, so we try to be as broad in terms of coverage and size at the molecular kind of phenotyping side, as well as systematic as possible from the phenome side, in order to try to infer links between genes, proteins, and disease on the largest class or groups of diseases, ideally across all clinical specialties. So this is why my talk, this is only to explain why, of course, some of the measurements that we have, if we go so broad, is not necessarily comparable with the precision that you have if you consider clinical assays. So the reason why I'm here today, or some of what has enabled the work that we have done, is because of the large advances in scalable technologies in proteomics. So here I've listed some that we've got some experience with. So on mass spec proteomics, is considered the gold standard. It lacks sensitivity for the less abundant proteins that are of interest. It measures fewer proteins, so peptide fragments. And why this is now more scalable, and we've done this in collaboration with Markus Ralser, who's at the CRIX, now biochemistry at BIH. It's not comparable yet in terms of the coverage of the proteome with these other two technologies that I state here. So this is Ohling's proximity extension assay, sorry for the typo, and this is SomaLogic's aptamer microarray. So these companies provide a much broader snapshot of the plasma proteome because they have affinity reagents that overcome kind of the difference in concentrations by measuring these at different dilution bins. So this is kind of an antibody-based technology compared to a clinical chemistry ELISA, and this is kind of uses short oligonucleotides or aptamers or somomers that really have an affinity to bind to the protein, which is determined by its 3D conformation. So it's important to keep this in mind, that when you have a measurement that uses this technology, actually it's not a concentration of the protein in plasma, but it's the abundance of the aptamer, and that's determined by its affinity to bind to the protein, determined by its shape. So we were lucky that early on, through collaborations, we've been able to do large-scale kind of plasma proteomics in this study. For the Fenland study is participants born and living around Cambridgeshire. And the reason why this study is useful for this purpose is they're relatively young and treatment naive at this stage. So mostly healthy individuals, no long-standing chronic conditions. So, in these over 10,000 people, we had genotypes. We did 5,000 aptamers using this now as a newer technology, 7,000, but this is what we had at the time. And we also used Olink. This was the panel-based approach now that's different to what is explored in UK Biobank. So it's 12 panels of 1,100 protein. Also did these are two small subsets, 500 individuals with these technologies, because we wanted to compare. And here you see the overlap. So not all, but almost all of the Olink proteins are also captured by SomaLogic, and this has enabled this early investigation of the kind of complementarity and comparability of these different platforms. So this is the summary slide of our kind of first proteome-genome-wide association study, or I should say studies, kind of over 50 billion tests. So we have these kind of 5,000 aptamers or protein targets, and used any variant. This is array-based genotyping, variants more frequent than 1%. And I think it's on one slide, you see probably work for 2 years done by Mike Pietzner and Eleanor Wheeler, summarized at once. So here you have the position of the genetic variant, kind of what you see on a Manhattan plot. And here you have the position of the protein encoding gene. And it's quite a lot of information kind of buried there. So if you take any one dot here, in some ways it's very simple. It's a single nucleotide variant, and it shows the differences in the abundance here of the aptamer at this particular position. And if that position is in or near the vicinity of the protein encoding gene, this will be pink and be on this line. And if it is further away, so this would be cis variation. And if it's further away, it's trans. It's a trans pQTL and would be indicated in blue. So this part is really simple. It's just lots and lots of tests. What you also see at this top here, this is the number of associated protein targets. And there are different examples. So what you see here, you see these lines that go up. For a single position, you see an absolute massive amount of association. So if this is a genetic, if this is I'm sorry. Stop moving. Apologies. Can you hear me? Yes. Good. So if this is a specific region of interest, and you find that it's associated with several thousand aptamers, that has some impact on what you think about its specificity on the protein target. So it's not just associated with a protein that it's encoding, but lots of other ones. And when you look here, there you see different regions that are hugely pleiotropic, and there are kind of different types of those, because when you see at this lollipop figure, there are some. The lollipop size indicates the number of independent variants at this particular locus that are associated with these proteins. So that distinguishes two different types of loci, some that are extremely pleiotropic, driven by very few variants, and others that are extremely pleiotropic, but kind of association dense, but driven by different variants in the same region. So that's kind of the summary of this study in a nutshell. And I don't need to read out the numbers, but we just detected a huge number of genomic regions that were associated with the vast majority of all of the proteins that were captured by the technology. Most of those, or many of those, had never been reported, and around 1,500 of those were in cis, so from the protein encoding gene affecting the aptamer. So why does this matter? Why do we bother? I mean, discovery is exciting, but could this have any utility? And this is one of the applications that I'd like to outline. So on the left you see this study that shows you by year the correlation between, from genome-wide association studies, of the number of participants included to the number of variant associations that have been reported. And there's not really any sign of slowing down, partly thanks to UKPGx and other amazing biobanks that have emerged. But for most of these studies, and actually when we looked to 21, there were 120,000 entries in the GWAS catalog. For most of these, the underlying causal gene or variant or mechanism is unclear. So there's a huge amount of follow-up work to be done, and that's the biggest, one of the biggest blocks to clinical translation is that we do not know the causal gene or variant. And hence, so because many of these are intergenic, we don't really know actually which gene to prioritize for follow-up. So one of the advantages of if you have a proteocentric view of the world or use a proteogenomic approach is, you determine, as I said, we take the cis-related variation, we know the causal chain from the gene to the protein. And if you find the association of this variant with the disease, that can help you to prioritize which gene in the region is actually responsible for your disease association. And so we did this systematically for where we could find overlapping data, and I should say so much of this is enabled by people generously sharing their genome-wide or genetic results, so that's really been a catalyst for so much of the work. So we're hugely grateful for that. So because we know the SNP in the protein encoding gene, this can highlight this, and we found for these that overall 25% of the cis-PQTs we had identified prioritized a different gene to the one that had been highlighted in the original GWAS study. And that's partly, I mean, if you find 100 loci, you're not going to go through great depths. Often people highlight the nearest gene or one that may seem biologically plausible. But if you think how much money and effort goes into these GWAS studies, this is a pretty neat way of prioritizing candidate genes for follow-up. So it's not the ultimate answer, but it gives you some way of prioritizing. And for another additional 14%, we refined a long list of genes to a smaller list of potential candidate genes. Now, we did this with a very basic tool where we simply looked whether the lead variant that drove our proteomic signal is the same one that's also the risk genotype for the corresponding disease. And that's kind of, as I said, a kind of quick and dirty approach, because actually it could be coincidental. The overlap could be coincidental because of LD. So here's a tool, a statistical tool called colocalisation that tries to kind of dissect this with more rigor, I should say. So you have this scenario where here you have the association pattern for a given trait. Let's say it's the protein, and here is another disease, so spondylitis ankylosans. And you really see from the association patterns this is distinct. It's nothing to do with each other. Even if the lead variants were the same. But here it's different. So here you see this association pattern really is related. It points at a shared genetic cause. And if we, like in this case, in our case, know here's the gene, here's the protein, and then the disease, we have kind of a causal chain of events. Of course, and we can infer a direction. Whereas if you had two traits that are two complex traits where this is not necessarily the case, let's say you take adiposity and insulin resistance, even if you identify a shared signal, the causal chain of events is not necessarily clear. You don't know which of the traits drives the other, but you may still be able to find the shared genetic signal. So I want to give you an example for what I've just said, where you can refine a causal gene. And this is based not just on taking here, so this is the association here, shared genetic signal between ISPO1 levels in blood, or the aptamer that's targeting this protein, and endometrial cancer risk that we identified. And this locus had previously been assigned to a different gene because there was an EQTL. And what you see here, what I meant with the colocalisation, so you see not only is the same lead variant for our protein association and the disease, but the signal in the region, the whole signal in the region kind of converges. So it's a shared genetic cause. You get a posterior probability for this, and that's very high. And it's also very high probability that this is the driving causal variant. And so then we identify a gene. We know that R-spondin-1 upregulates estrogen receptor alpha, which is implicated in endometrial cancer. Also, there's evidence in the literature that a knockout mouse has abnormal development of reproductive tissue. And so while this is no proof, it helps to identify or prioritize a potentially causal gene for follow-up that has refined what we previously implicated with no extra effort. So through the integration of these different omics layers, so from genome to proteome, or in other cases, expression data, as I'm going to show you now, 2 different phenotypes. So I want to come back to what I said earlier. So this is the distribution of the SomaScan kind of protein targets, and you see that the vast majority of those that we detect, almost 70%, are not kind of actively secreted and hence, but still detectable in blood. And so we used a trick because there's the GTEx consortium that has gene expression data across 49 different tissues and has done genetic analyses of these. And again, all of these data are publicly and openly available, which is amazing. And also other data sets for whole blood and so on have become available. And so we systematically tested for all our cis-pQTLs whether or not there is a shared signal between the expression of the gene across different tissues with the levels of the protein in blood. So, and the reason for this is that this can help us to identify, if this is specific, to pinpoint the tissue of origin where this may actually have originated from in plasma. And so if we do this, this is just a summary plot, which is not very legible, but here's the summary that 50% of the cis-pQTLs we had identified showed strong evidence. We had a very high level of posterior probability to accept this, 80%, with a cis-eQTL or a splice QTL. And there were 145 of those were tissue-specific, where that really then helps you to infer where is the tissue damage potentially that has led to the leakage of the protein into plasma. Now we can use the same technique that we've just used for gene expression across these 49 different tissues to now think about diseases and disease associations. So we use the same trick here. Oh, sorry. Wrong button. So here's the genomic location. This is the signal for the protein. You could imagine this was the expression of the protein in one of the tissues. And then we now integrate information across all of the diseases where we have access to the genetic data and integrate this for this specific region of the protein encoding gene. And that's exactly what we've done with this statistical technique. And the example that I want to give you is a different paper that's not yet published. And this is related again to the pandemic. So the COVID-19 host genomics initiative has also publicly released their results. They've looked at different definitions of outcomes. One included infectiousness using different controls, but also COVID severity and prognosis, so risk of hospitalization and a more severe course. So we analyzed all of our cis-pQTLs across these various COVID definitions. We confirmed what we and others had previously published about ABO and, the other one escapes me now. Anyway, so we did find there was previously a reported peak on chromosome 11, and we found that L5 was the most robust candidate protein for this particular peak. And if you look, and it was most associated not so much with infection, but with severe outcomes. And here you see exactly what I've described to you before. Here's the genomic region, here's L5. And what you see here is, number one, that it co-localizes with a cis-eQTL specifically in the lung, in GTEx tissues, so none of the other tissues. So here's the same signal, same lead variant for lung. And then here you see the different definitions of the initiative showing the same peak with the same lead variant. And the odds ratio for severity here, for hospitalization, was almost 5. So it's a massive effect for this one variant, common variant. And you can also do something where you do this trick of colocalization across different, different entities or modalities, so expression as well as these different outcomes. And this came out as most likely to be the causal variant. And then in collaboration with colleagues at the Berlin Institute of Health, they used single-cell RNA-seq to determine L5 expression at different sites of the respiratory system that they had access to because of studies that were already ongoing because of the COVID pandemic. And they found that L5 was highly expressed in epithelial cells that are also the target of SARS-CoV-2. And furthermore, they found that L5 expression in cystenticular cells correlated with TMPRSS2 expression, which is a transmembrane protein that facilitates viral entry. And these cells are the epithelial cells in the kind of nasopharynx, so the entry of the virus. So using totally agnostic, kind of broad approaches that systematically integrate these outcomes and look across all these proteins, we were able to find an association that is incredibly specific, and for a genetic association, relatively strong in its effect on the adverse prognosis of these patients. Now, the next step really is to see, this is still disease-specific, certain infection and the prognosis for this disease. But actually the ability exists, as I've just said, to look across all diseases that you have access to in terms of the genetic data. And that's the next thing that we did, and this is kind of what we call the proteogenomic map of the phenome. So literally just to integrate the data across all the different diseases. And if you do this, then you see here on the left, it is ordered by here diseases and continuous traits, and you just see the number here, actually the number of co-localizing proteins. So you see the diseases where there's a lot of proteins that you see a shared signal with listed here. Then on the right here is the exact opposite. So here you have the proteins ordered by the number of disease associations that are associated with a single protein. And this includes some of the very known pleiotropic genes, such as GCKR, but also includes some other examples. I just want to highlight one in the interest of time today. So if you graphically present this now, I thought I've advanced. So if you graphically represent this information now that I've just shown you in the other figure, then you end up with this, which is what we call the map. So this is over 1,800 connections between these over 400 protein targets and these over 500 curated phenotypes to avoid redundancy in these diseases. And so here I've just pulled out the kind of fibrillin-3 centered subnetwork. So this is one example from this network. It is one of the most extreme examples, that's why I've chosen it, but nevertheless. So here's this one protein associated with 37 different conditions, all kind of soft and connective tissue conditions or anthropometric traits like fat and height, fat content and height. So that was quite intriguing. And if you look here now at this is kind of extracellular matrix glycoprotein that's encoded by this gene, FMP1. If you look here, this is again this stacked kind of stacked regional plot. Here is the expression of so this is kind of lower levels associated with this kind of A allele, lower levels of this protein in plasma, lower expression of the gene in adipose subcutaneous tissue, and then risk of diseases all associated with kind of what we called abnormal elastic fiber morphology, so hypermobility, hernias, varicose veins, and so on. And the converse, lower risk of conditions such as carpal tunnel syndrome, so kind of consistent. So again, in a totally hypothesis-free approach, you can identify examples where you link a specific gene and protein to a kind of total mix of kind of etiologically probably related diseases that have not been brought together and across different clinical specialties. So in terms of pharmacogenomic approaches, this of course gives opportunities, and while this may not be the one where there's an existing drug that could target this, but if you think about the specificity of associations or the potential applications of drugs, this can really help to prioritize. So I'm coming to the end. So I just wanted to say, for those of you who are considering doing proteomics in your studies, we have done a lot of work on the comparison of different technologies, and that's published. If you're interested, not just kind of from a measurement or observational perspective, but also on the impact that the technology or the platform has and the consistency of this on the disease associations that you see. So that's published there. That's a slightly more technical paper. And then also, here's one of my colleagues from Cambridge, Mina Kopula, is currently working on actually bringing together the data across all of the studies and looking forward to UK Biobank having the Olink proteomics on separate meta-analyses of the different technologies, both Olink and SomaLogic, to enable a much, much broader cross-platform comparison. Because if you're interested in a specific protein as a drug target, ultimately, and if the platforms deliver comparable results, you don't really care who or how it's been measured, but you do care about bringing it together to have the best and most powered genetic discovery. So that's new and ongoing, so watch this space. So we've also developed with colleagues at the Helmholtz Center in Munich, led by Gabi Kastenmüller, a kind of a web application, so where you can explore dynamically targets, explore the network, look up SNPs, and actually we're just enabling kind of a dump download of all the results from this study, and also other studies are on there as well, on metabolites and so on. So I hope, in summary, I've shown you at least the potential of these omic technologies for translation and the understanding of disease mechanisms, the identification of disease-specific, as well as kind of shared genetic causes. And none of this would have been possible if it hadn't been for successful academia, industry collaborations, both from the kind of measurement side, but also in the funding of some of these endeavors. And I haven't talked about prediction, but actually that's one of the efforts that's currently ongoing for proteomic prediction. And again, as I said, I think it's very important to compare to the best possible benchmark that currently exists, so over and above what's currently done clinical. So that's the next endeavor. As I've mentioned, the cross-platform GWAS, mentioned the prediction, and of course, ultimately, in terms of prognostic stratification, this has enormous potential once done at scale, with sufficient numbers of patients to look at adverse events that occur. So with that, I have to thank very many people, and I have the privilege of presenting the work on their behalf. So I'd like to thank them all. I hope I've named all the individuals most relevant for the work I presented today. And then finally, thank you all so much for your attention. And if you like what you saw, we're currently hiring. So if you're interested and computationally minded, please email me. Thank you very much.